Hello students! Tonight we are going to wrap up Chapter 2.3 by discussing active transport as well as endo and exocytosis. We will continue to use concepts such as selective permeability, concentration gradients, and transport proteins that were introduced in parts 1 and 2 of this section. So, let's begin by recognizing that sometimes a cell has to move molecules against their concentration gradient. Another way to state this is moving molecules from areas where there are fewer to an area where there are more molecules. This is, as you might notice, the opposite of passive transport. In other words, it is like trying to swim upstream against the current instead of calmly floating downstream with the current. Some examples of situations in which this might occur would be moving nutrients or glucose or salts into a cell that already has many of these molecules stored in it. All right, so let's look at the first picture here. First, we can identify that a concentration gradient is present. There are more solute molecules inside than outside, so the direction of the concentration gradient looks like this. In this situation, the solute molecules would naturally want to move this way with their concentration gradient. However, let's say that these are important calcium ions, and your cell wants to acquire as many as possible to store and use later. In this case, the cell may want to take these solutes that are remaining outside the cell and move them in, which, as you can see, is against their concentration gradient. Okay. So, since this is not the natural direction for molecules to move in, some energy is required to make it happen. This type of transport is called active transport, and it uses a molecule called ATP for energy. We have mentioned ATP before when we talked about mitochondria and how they convert food into energy. And we will talk more in depth about this important molecule, ATP, in the next section. All right, finally, notice that active transport of solute molecules requires the use of special transport proteins. In this diagram, this here is the transport protein. The reason that transport proteins are required is because the ATP is actually used up by the transport protein to provide the protein with the power to pump the solutes against their concentration gradients. All right, finally, some molecules are just too large to pass through the cell membrane at all, even with the help of a transport protein. The processes of endocytosis and exocytosis are used to get the largest molecules into and out of cells. The first of these, endocytosis, is the process by which a cell takes in a substance by surrounding it with cell membrane. This process is illustrated in the first panel. First, a small part of the cell membrane, highlighted here, folds in like this. So this is one. And The reason for this is it's going to create a pocket for the large molecules that it wants to move into the cell. The pocket will then deepen here until it eventually pinches off. Okay, so here you can see the whole piece of the cell membrane that we use to surround the molecules is pinched off. And it is going to form a little packet of molecules, which we see in here, number 4. And this packet of molecules is called a vesicle. Okay, and this vesicle can now move throughout the cell and deliver these important large molecules wherever the cell needs them to be. The opposite process, called exocytosis, is a process through which vesicles release their contents outside the cell by merging with the cell membrane. So this is shown in the bottom panel. And as you can see, it's just about the opposite of endocytosis. You can see here we've got this vesicle containing large molecules. It will move towards the membrane here until it merges with the membrane. And when it merges with the membrane, it actually opens up and releases all of those large molecules outside of the cell. All right, that wraps up our discussion of the types of molecule transport. In these 3 sections, or I should say in the 1 section, but the 3 parts, you have learned about passive transport of molecules, including diffusion, osmosis, and facilitated diffusion. And you've also learned tonight about active transport and endo and exocytosis. We will continue to review these concepts in class tomorrow, so please make sure you review and complete the Quizmodo tonight. See you tomorrow!